My name is Astha Symes, and welcome to Live Longer World. In today's conversation with Mike Levin and Pamela Lyon, we hit the ground running. So I just want to cover some basic groundwork on how they view cognition. In her theory on basal cognition, she states that we don't have to view cognition as something that's necessarily associated with just the brain and the nervous system. In fact, in organisms that lack a nervous system, she shows through experiments that they still have some of the properties that we associate with cognition, namely memory, decision making, and more. So, what does this imply for how we think about cognition? Can cells, tissues, artificial intelligence all have cognitive power? And what does this mean for regenerative and longevity medicine? This is something we dive into the podcast. So I hope you enjoy the conversation. Pamela and I were talking about this offline as well. And I think a good place to start would be understanding how you got into the field of cognition, because it explains a lot of your work on cognition research. And I believe it was you doing a PhD in something else, and you stumbled upon the fact that the current models of cognition are not correct. So would love to understand how your foray into cognition research, and then also what you think is incorrect about some of the current theories of cognition.
Okay, thank you. I started out in a PhD doing a cross-cultural comparative analysis of 4 Buddhist propositions. This is Buddhist philosophy, right? Now, behind those propositions is a concept of mind, cognition, what mind is, what cognition does. And I realized that I actually couldn't do the comparison unless I could come up with consensus views of what cognition was on both sides, on the Western scientific side and the Buddhist side. Now, Buddhist side, no problem. They had a very, very clear understanding of it. They'd been working with it for 2,500 years. Meditators had been developing extraordinary states of mind using this concept. But when I went to the Western side, I couldn't find anything. Now, people say that's really uncharitable, but what I was looking for was something that you could do something with. That you could actually say, well, this is the way the mind works. This is what it is. And it was like the mind is the brain. Well, the brain does a whole lot of stuff other than just being a mind. The mind is computation. Well, what does that actually buy you when you're trying to figure out what a mind is, what it does, what it's for, all that sort of stuff? It didn't seem like that there was enough there. And at the time I was looking at this was cognitive science. There was really very little biology, almost no biology. And evolution was kind of trotted out as as something to indicate that you know something about biology, but it really wasn't pursued in any way, shape, or form. Now, understand at this point, in the late 1990s, coming up to the 21st century, people were only just beginning to talk about embodied cognition. They were starting to talk about animal minds, animal cognition. I mean, the seminal book on animal cognition was published in 2001. 2001. That means there was a cognitive revolution going on for practically 50 years before we got to that point. And I was dumbfounded. I didn't understand how we got here. And I couldn't find anywhere a kind of a T-shirt worthy definition of cognition on the Western scientific side that I could compare with the Buddhist one. There just wasn't one. There were lots and lots and lots of them, lots and lots and lots of them. So I thought, oh my God, this is actually the problem. This is the problem. Forget the Buddhist propositions, forget cross-cultural comparative analysis. This is actually the gap. This is the gap. And so for me, the question was, how on earth do you go about filling this gap? And so for me, it meant it has to be based in biology, because it has to be based in evolution, because we know that living things started out small and they developed over time. And there was lots and lots and lots of debate and argument about where on the living spectrum cognition kind of jumps out, right? Genuine cognition. And I thought, I can't get involved in those arguments, because I think the basic premise of that argument is wrong. It's not, where does it jump out? It's like, what is it doing? What is it doing for an organism? You have to ask those questions first. And so I did a quick dash to the microbiological literature just to see what in the hell was going on there, because I had no idea, and I'd been told by everyone I was in discussion with that there was nothing going on there. It was all just reflexive, hardwired, programmed, all this sort of stuff. Nothing to see here, right? So luckily, I had been trained by my experience of writing about the molecular work of two Nobel Prize winners in the year that they won the Nobel, Joseph Goldstein and Michael Brown, who won for cholesterol. I was working in the publications office of what is now Southwestern Medical School. They won the Nobel. Somebody had to write about what their work was, and it was down to me to do it. And when I went to ask them questions, they handed me a stack of primary material and said, "You're a smart girl, come back with questions." And I just went, "You're kidding, right?" But as it turned out, I took to it like a duck to water, and I came back with questions, some of which were open questions in the field. And they said, "That's a very good question. We don't have an answer to that yet, but there are people looking at that." And they gave me confidence to do it. So when I went to the microbiological literature, I had already been trained to go to that kind of literature and kind of work it out. And what I found was just dumbfounding. I mean, just dumbfounding. Not only sensing, but sensory motor activity and memory and non-associative learning and making decisions under uncertainty and error correction and anticipation or prediction. It was like there's communication. There was an entire world there that nobody was touching. Nobody was touching. And everybody thought there's no there, right? So once I discovered that there was something really, really basic that involved cognition, I just said, okay, well, what is biology? What has biology got to say about cognition? So I started looking at self-organizing complex systems, that entire body of work. And I was looking at, I looked at Maturana and Varela. I looked at Stuart Kaufman. I looked at Walter Elsasser. I looked at Robert Rosen. I looked at Ludwig von Bertalanffy. I have this strange mind. I kind of hoover up everything and then feed it to myself, and my subconscious kind of runs the numbers and comes up with something. I really don't know how it works. And so I started seeing patterns in self-organizing complex systems. And that gave me the first set of what I called biogenic principles. It's like, and when I say principles, it means whatever explanation you come up with for cognition, it must accord with these principles. And what I found in cognitive science was that there really weren't very many principles that held anybody to account. People could make up almost anything they wanted to and say, well, I've got a thought experiment that says blah. And dozens and dozens of papers would be written on blah, but it actually didn't move the dial at all on what cognition actually is and what it does and that sort of stuff. And so it wasn't that it was wrong. It's just that there was an elephant in the room that nobody was looking at because everyone was concerned that any definition of cognition had to apply to machines as well as living systems. And Noam Chomsky wrote like a brilliant takedown of this entire ethos in an essay on the 1990 side of the turn of the century. And when I read that, I just went, right, that's exactly what I'm seeing. That's exactly what I'm seeing. And so I was constantly triangulating what I was finding with people who were leaders in the field to make sure that I wasn't saying something really, really, really off. And that's because I used to be a journalist. And when I was a newspaper journalist, everything I wrote had to pass muster with people who read it. And all kinds of people read stuff in newspapers. Well, they used to. Anyway, we used to have a more common view of how the world works. And I was just paranoid about being wrong. I always, as we say, I covered my ass really well, because I had to. And that's how I got into this field.
So Mike, actually, question for you. When you think about some of these self-organizing complex systems, and the biogenic principles and cognition, does this also apply to, say, anthrobots and xenobots and cells and tissues and some of the work that you do?
Yeah, absolutely.
Yeah.
And honestly, I mean, and this is probably a discussion in the end for another day, but my view is so I agree with everything that Pamela just said. There absolutely is some weird things going on with these systems, but I actually I take it weirdly further because we are now finding some of those same things happening in very minimal systems that nobody would call
Yes.
alive.
Yes.
And I think that these vitalist / organicist thinkers are on the right track. Absolutely. I just think they stopped too early. I think they didn't follow their convictions far enough because they really want to. And I get yeah, I'll just say I get yelled at way more by that crowd than I do by the molecular biologists. I mean, they don't like the cognitive stuff that we're saying about gene regulatory networks and whatnot. But they're actually less mad than the other folks because it's pretty important for a lot of people to keep a very strict separation between the majesty of living things and the machines that they are remembering. These boring sort of mechanical things. And I think they should take those views and put them on steroids because I see this stuff going all the way down. We're just not very good at noticing it in some of these weird, like really minimal systems. You have to pay a lot of attention to find it. But yeah, I mean, but anthrobots, xenobots, yeah, sure. I mean, those. I don't think you have to be too too imaginative to see it there. I mean, it hits you in the face when you're watching these things. The creative problem solving that they do. I mean, you see it. I would love to hear. I mean, I know and by the way, I first came across Pamela's stuff in 2006, the biogenic approach to cognition, which I was just like, I thought it was just amazing. And I would love to hear just a few of your favorite examples from the microbes, from like microbial war. Like, I mean, you ran through some, but like, what are some of the amazing things that you found in that world?
Okay, well, thank you very much. And why didn't you reach out in 2006, for heaven's sake? You didn't contact me until 2015.
Yeah,
The
I don't
cognitive
know.
cell.
Well, yeah, I was. I'll tell you why. I don't know. I don't know exactly why, but roughly speaking, I wasn't reaching out to almost anybody at that time. I had just
Yeah,
started. I had just
yeah,
started my lab. I was trying
yeah,
to get the bioelectric stuff
yeah,
off the ground. I
yeah,
was kind of,
yeah,
keeping fairly focused, and it
yeah,
was some
yeah,
time before
yeah,
we got big enough that I could start
yeah,
branching
yeah,
out.
yeah. Yeah. No, okay. Okay. Bacillus subtilis is now my current favorite microbe, right? Because in biofilms, first of all, they are such a wonderful example of bioelectricity and how bioelectricity is used communicatively between individual living cells that come together as a collective. I I love that example. I love the fact that 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 the centers of biofilms, when they start to starve, they send out an electrical signal to the periphery, which basically says, stop eating and reproducing and let some of those nutrients diffuse into the center so we can eat too. And that they basically time share scarce nutrients, because biofilms only form under some issue around declining nutrients. That's it's a well, that was the original view of them. I'm not sure that's true anymore, actually. I think that it's I think that it's a good survival strategy no matter what you're doing. But but bioelectricity is also used for different colonies of Bacillus subtilis to timeshare scarce nutrients between each other, and which is really amazing. So I love Bacillus subtilis. But the first microbe that captured my heart and still has a huge, huge, huge role in my thinking is Myxococcus xanthus. Because first of all, it's a golden microbe, and it is slow. It is slow. It can't do much on its own. It is relentlessly social, and it's predatory. It is predatory to the point where C. elegans, which is so much bigger than these single cells, if they sense that Myxococcus is around, they will flee. They will go somewhere else. They avoid Myxococcus xanthus because they are such profound badasses. They collectively they can prey on colonies of other bacteria, but they also can prey on larger things, which is kind of weird. And they do so by coordinating their activity, getting them surrounded, and then releasing enzymes that will break apart cell walls, which means the guts of the cells start breaking out. And C. elegans, their cells are not as highly protected as bacterial cells are. So breaking apart a bacterial cell wall is a harder ask than something softer. When I found out that C. elegans were afraid of Myxococcus xanthus, I was just like, how is this even possible? And what Myxococcus xanthus does when it feeds, like on a colony of something, it engages in this oscillatory behavior, which basically sends nutrients back further down the line so that these cells are also feeding and reproduce. And so more and more and more, their colony gets larger. And when there's a step down, we used to think it was starvation, but now it's just a step down in nutrients. When nutrients were like here, and now they're reducing, they start aggregating, aggregating into fruiting bodies. And when they aggregate, in one of the best descriptions I ever read by Shimkitz was, he said, it's like the great animal migrations of the Serengeti. It's like you've got tens of thousands of cells aggregating in herds that then come together to form these mounds that then differentiate. And in order to form the mounds, all of them have to be competent to create these structures. And some of them aren't. And somehow the cells that are competent can transfer their competence across the cell membrane to make them competent. This is called complementation, which the mere existence of which made me start questioning all of the everything that I'd read about the evolution of cooperation. It's like, hang on, if this is happening at the microbial level, cooperation is really, really built in at the bottom, and because you can sheet this home to kin selection to a certain extent, but in the Bacillus subtilis cases, they're much more diverse, they're much more heterogeneous than kin selection by itself would suggest when they look at biofilms that are grown from wild populations and stuff like that. So there's a lot more going on in the living world than we know. And then so when they build these structures in which at the top you have mixospores, so there's a lot of differentiation going on, right? And sporulation is just massively energetically costly. So there's a certain proportion of the cells that they will lyse themselves to provide nutrients to keep this going. But then after the fruiting bodies are formed, there are cells that remain as sentinels to keep the fruiting bodies free from predation. Now, how on earth did that even, and now I will tell you about something that Jim Shapiro described as the most interesting paper that nobody's ever read. And that is by Martin Dworkin, who was the one who tamed Myxococcus xanthus for laboratory study in the first place. He was just, as you do in the lab, as you know, you kind of messing around in the lab and seeing what might happen if you do this. They put glass beads, inert glass beads, no chemical signal whatsoever, onto a medium in which Myxococcus xanthus was moving and growing and moving. And Dworkin, this was a 1983 paper, Dworkin and his colleagues observed Myxococcus xanthus migrating to the glass beads, crawling all over them, and then leaving. What on earth were they migrating toward? There was no chemical signal. They did this experiment over and over again to make sure that there was no chemical signal. And on that basis, Dworkin mistakenly assumed that what Myxococcus xanthus does actually isn't chemotaxis. They do chemotax. But based on this experiment, he said something else is going on here. But the only thing that he could, I think it probably has something to do with the deformation of the medium, that it's it might be a mechanical signal. It might be a mechanical signal, but that means Myxococcus xanthus has more sensory modalities that mean something to it than we had any idea that they do. So its Umwelt is richer than we knew. Okay, but when you put this entire picture together, the Umwelt of Myxococcus xanthus is just extraordinarily complex, because there are all these signals that do things. There are all these signals, many of which we still don't know what they do. And so that's my favorite microbe. even today, even though we don't know how bioelectric it is yet. I'm sure it is. And I'm sure that the collective fields of Myxococcus xanthus are really, really important in its collective behavior. I have no doubt of it. I have no doubt of it. And all of that's due to your work, my dear, which changed my view of everything. I used to be a very dedicated, oh, we can't go down to the genetic level. That's just taking it too far down, because I just thought, I mean, if we do that, then we are vitalists, basically. We are vitalists. And I had been trained not to go there. Do you know what I mean? But once I started, I had all the way through, I had seen evidence, because I came across Ladislav Kovach's work and on cognitive biology, and that led me to Brian Goodwin. And now that's a completely different area, and we can talk about that, and I'd love to. But that convinced me that you had to be right. You had to be right. And now I have absolutely no doubt that you're right, that it goes all the way down to the molecular level. We have no idea how any of this works, why it does this. But I think that we have to look to self-production. You know what I mean? Autopoiesis. Autopoiesis kind of demands it. And once you get a system that is self-producing, not just self-organizing or self-maintaining, I don't think that we can do without it. I think these kinds of, I'll say scare quoted cognitive, but it's cognition, okay? All the way down. It's cognition all the way down. And the sooner we realize that, the more, I think the sooner we realize that, the brakes will suddenly be off a lot of science, and people won't have to dance around and caveat this and that because they're worried about the V word. Do you know what I mean? Let's worry about that later. Let's just put it to the side and say, we'll deal with this later, because it's actually not important. What's important is understanding what's going on in these systems. That's what's important.
Yeah. Your point about the beads and no chemical gradient. We had this Neuroshim Murugan, and my group did this. We had this Physarum, the slime mold. You put it right, and you put it in the center of a petri dish. We have these little tiny little glass discs. I think they're about 10 milligrams a piece, but they're just glass. There's no chemical on them. And you put 3 over there, you put 1 over here, and it kind of grows out a little bit for about 4 hours. It tugs on the agar, right? You can watch this little, and it turns out that all that time it's building up information about its environment, and afterward, boom, it just shoots for the 3.
For the 3
consistently, right? Consistently,
consistently
it goes for the 3. No chemicals, no gradients. It just from that, and the sensitivity is insane, because these things, we did all kinds of stuff to try to fool it, right? So you can stack the 3 on top of each other, and you can you can put you can rock it, right? We put it on a rocker, and we did all these things to confuse it, but it's basically it's sensing the strain angle on the order of 10 centimeters. It's this large thing, and it has this like idea of what's going on around it. It's amazing, this idea of this Umwelt and these things, pulling together a representation of what's going on before it acts, long before it actually does anything. It's doing that.
Oh, one more thing about Myxococcus xanthus. This is another rarely cited paper that I absolutely love, is that there is evidence to suggest that Myxococcus xanthus will extrude CAMP as a signal to draw E. coli into its orbit, because it's too slow to follow the flagellated bacteria. It's too slow, so it needs to lure them in close so it can then surround them, lyse them, and eat them. I mean, a microbe, emitting a biologically relevant chemical as a signal to draw in prey? Give me a break. You know, we just really don't know what the hell's going on in the world. You know, our view of what the living state was like, and these really, really simple instances of them. That's why the xenobots are so important, because they show really clearly what's built into the system. And it's not all in the genome. I mean, the genome's the hardware, it's the software that runs the damn thing, which is why all of the work that you've done on bioelectricity is so really important. And I will say again, when I first the first time I heard about it and I heard you explain it, I just went, my God, this guy's going to win a Nobel Prize. And I knew him when, you know, and I know you're not supposed to even think that way, but seriously, the whole bioelectric field that's opened up, you know, with you basically sticking your neck out on the chopping block and letting people take, you know, shoot arrows at you and try and cut it off and all that sort of stuff was absolutely fundamental to our understanding of a really fundamental biological capacity. And I am so grateful for that work. So grateful for that work. Thank you.
Wow, that's amazing. And yeah, and I mean, there were a number of, I mean, I got my inspiration from people like Harold Burr, you know, who in the 1930s was talking to, right, with he had nothing but a good voltmeter. That's all this guy had, and a crystal ball, apparently, because just with that,
and a crystal ball.
And a crystal ball. No, it's crazy. It's amazing. There's a few other examples of this, of somebody who sees the future so clearly with very limited actual, you know, ability to really prove any of this stuff. You know, he went around and he just measured things. He measured trees and embryos and you know, psychiatric patient skin potentials, and and on the basis of all of this, he in his little book, he laid out pretty much we're still, you know, I'd say we're probably no more than 3/4 through all of the things that he found that we've actually now documented. But he was just, you know, sort of visualizing all these things, you know, many decades into the future. And there were others, you know, people like the Lionel Jaffe and all of his folks, and they caught so much crap in their day from all the molecular biologists. It was amazing. Oh.
Thank you for that example, because basically what you've just described is that the creativity of the human mind interacting with nature directly is really essential to our understanding.
Yeah, that's exactly right. And that's, you know, to me, and I'll ask you towards the end what you think the future of all this is. To me, one of the biggest open areas is the creativity aspect, because because we can see there are parts of what these things are doing that are algorithmic, that are turn the crank, you know what the process is, you turn the crank, you get your answer. And then there are other parts that there is no better way to explain it than creative, some kind of creative process. It is not, it's not algorithmic. There are no steps that you know how to do to you know, to solve some of these things, and that's, I think, baked into the bottom of what we're all interested in, and that is to me that's the big open frontier.
Well, I think that you've been dancing around this a bit, and I've been dancing around it a bit, and I think that when you look at what it takes to be alive, to maintain life, that whole self-production thing, autopoiesis plus self-organization and self-maintenance and that sort of stuff. This is why we have so many responses to stress. Stress responses are, I think, on one level, the engines of creativity, because you have to get out of this problem. Now, in microbes, once again, provide a very interesting entree into this, and that is there are these factors called ECFs, which unfortunately it's been so long since I've written it out that I can't remember. It's environmental something factor, and we don't know exactly what they do, but they seem to be a kind of a wild card that come into play when kind of an overwhelming stressor is encountered. And it is believed that they provide some degrees of freedom, that the standard stress responses that are kind of built in to the genome and the hardware and software of the cell, they're kind of wild cards. And now that may just be because we don't know what they do and they seem to activate a lot under stress. But they're an additional tool for dealing with stress. And I wrote a chapter years ago that nobody read really, but that's okay. That happens in chapters in books. They don't get read. Papers get read. And that was, I was advocating for a coevolution hypothesis of cognitive capacity and stress response, that these two things ratcheted each other. Yeah. They scaffolded each other. And the thing that convinced me of this, first of all, was in microbes, the most sophisticated, cognitively demanding behavior they engage in, it's all stress related. It's all stress related. It's all in anticipation of stressors that are existentially challenging. And then when we started recognizing in the early part of this century that the brain really wasn't an immune privileged organ, that immune system elements like cytokines were working everywhere in the brain. And not only that, they weren't just activating under stress, they were involved in normal memory, normal learning, normal cognitive activity. It's like, what do you mean tumor necrosis factor alpha is involved in the scaling of synapses that lay down a memory? What the hell's that? What do you mean IL-1B and IL-6, which are notoriously, heavy duty actors under stress conditions? What do you mean they're involved in normal cognition? That doesn't make any sense unless you start looking at what it takes for something to live. And all of the gigantic things that can go wrong. I mean, really, a lot of them are predictable, but so many of them aren't. Right. And so I think that stress responses are the source of our creativity, actually. And I think that they had to be in the very beginning. I mean, like, just think about it. How on earth did we get photosynthesis when exposure to UV radiation breaks DNA? Do you know what I mean? Think about it. Think about it. You have to be close enough to the sun for long enough to photosynthesize, but you have to be able to protect yourself against DNA breakage, which is the one thing that's going to make sure that you can't reproduce effectively or reliably or whatever. I mean, just the evolution of circadian rhythmicity, of partitioning physiology so that it can be done at times that aren't dangerous. I mean, just there's so much going on early on in the origin of life. And And, when in the hell do ion channels show up? When do they show up and how do they show up? And what do you mean? Okay, so we're pretty clear now that stromatolites, fossil stromatolites in Western Australia and other parts of the world date back to 3.85 billion years. But what in the hell are we talking about when there's basement rock off the coast of Greenland, your homeland, that indicates organic life at 4.2 billion years. I mean, what on earth is going on here? Because 4.2 billion years, I mean, we are being pummeled by stuff from space. There is water on this planet that is older than the planet. Do you know what I mean? It's older than this planet. And how did that get there? I mean, it's all coming in from space. It's all coming in from everywhere. And we've got Hadean conditions. Do you know what I mean? Which means Hades, not nice, not really peaceful and easily synthesizing life. What on earth is going on here? We haven't got a clue. But the good news is we haven't got a clue. So there's this entire area that really needs exploration. And until we can get a kind of a handle on what are the basic kinds of functionality, that you need to keep the show on the road, so we can even sort of begin to trace what might have been the original cognitive contribution to this state. Until we can get there, we're not going to understand the rest of it. We're not going to understand the rest of it. And because things had to be sensed pretty damn quickly in order for these things to be partitioned, for circadian rhythms to evolve, for understanding, and I don't want to use the word understanding, apprehending that proximity to a particular kind of wavelength of something is going to damage you, such that you need to move away from it, when it's really, really high. Or you've got to have other protective modes of being, which is why I think, there is a program, already extant on the planet called Major Transitions in the Evolution of Cognition. But most of that starts at nervous systems. We're not talking about the basement level of all this stuff. And until we understand the basement level and the kinds of transitions that we're looking at, and the development of mechanosensitive ion channels are really critical in that, really critical. So, yeah.
That's fascinating. Mike, I'm curious, how do you think some of this ties into your work on stress sharing as a cognitive glue?
Yeah, I was about to say, I don't know, Pamela, if you've seen this, but we've got some stuff. I mean, I agree 100%. Stress is critical. We've got now a paper on stress sharing as in particular a mechanism for binding competent subunits into larger whole.
Will you
larger.
send that to me?
I will when we're
send that to me. Yeah,
I absolutely will. But we also have so there's that. But we also have experimental work, which is not published yet. We're still kind of in the middle of it, looking at how evolution has reused some of the molecules that initially were about failures to protect your DNA, protein misfolding, all of those things. They got repurposed for these large scale things like, hey, my anatomy isn't right. And there's some amazing stuff on the role of stress in binding these anatomical intelligences, that are trying to keep their navigation in morphous space correct. So yeah, I agree. I 100% agree with you. But even that you could take all the way down, and I say these things and then people kind of laugh, but geometric frustration, is
geometric frustration.
no, well, it's a term, right? So when you have,
I love it.
right, but my so that's a term from physics and math is geometric frustration,
yeah.
and I keep saying it's real frustration. It's
It's
actual real frustration
Yeah.
because what they have is these, like, if you can think about either tiling the plane with certain shapes, and then eventually you can work yourself into a corner where it doesn't fit. If you make it fit, then it'll somewhere else won't fit, right? Or when you have magnetic domains, these icing models where they try to make everybody aligned, but somebody's not aligned, and if you fix that one, then it propagates somewhere else. Well, this inability to get all your parts onto the same story of what should be happening, right? To convince all your parts to work towards the same, to buy into the same vision of what the hell you should be doing is actual internal frustration, and it drives a lot of downstream steps. And I think that's exactly what evolution capitalized on in the kinds of things that we think of, where we're internally incoherent and all this. I think it goes all the way, as we said, all the way down.
That's a wonderful concept. I love it.
Yeah, Richard Watson and I've been doing a lot on this now, thinking about, like, what does it take to get your parts aligned, and to bend their umwelt and their landscape so that they do the thing that they want to do, but actually it's serving this global purpose in another space that they can't they can't visualize, but you're bending their space in that way. Yeah.
Well, so nitric oxide is a really important stress product that became like the dominant second messenger in multicellular in a lot of multicellular life.
That's a good point. I should take a look. We haven't been tracking it, and I think they have sensors now, right? Don't they have nitric oxide sensors, like fluorescent reporters and stuff? I think they do.
They do, and the person's the work that you want to look at is coming out of Nicole King's lab. She had a PhD student who was doing, they call him Dr. No because of his focus on this. So some of the really interesting work is coming out of her lab.
Interesting. Okay. Yeah, I'll check into that. Cool.
Yeah.
Well, one thing I want to make explicit is that when both of you speak of cognition all the way down, it's not just that it implies that cognition is not just associated with the brain, which is how most people think about cognition. And I know both of you have co-authored a paper on this as well. So I'm curious, what do you think are some of the implications for this when you start thinking about cognition so differently, where it's not just about the brain, it's all the way down. You have cognition and some of these microbes and beautiful examples that you gave, Pamela. What do you think are some of the implications when we start thinking about it in the future?
Mike, do you want to go first?
No, please, you go.
Well, as I alluded to earlier, but didn't specifically say why, there are all kinds of things that we are finding at the molecular level that people recognize. They try to avoid language that is cognitive because they know they'll get jumped on. Right? But what they're actually looking at are cognitive phenomena. And if they could recognize that it is cognitive phenomena, not like cognition like we've got, because ours is very specific to us and it's very special and all this and blah, 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 blah. But if they could recognize it in a bare sense, this is seriously cognitive. They can ask different questions, they can investigate things in different ways, and they can develop theories that they couldn't develop before. And right now, biology has always been criticized as basically descriptive. And you're just you're describing this phenomenon, you're describing that phenomenon, you're describing that phenomenon, and it's like bean counting, right? And there's not enough glue between the beans, right? That will hold them together. Well, as Mike said in one of the titles of his papers, There is a cognitive glue. There is a cognitive glue. And if we can start recognizing that these are phenomena that we recognize in ourselves, that we can see them across the spectrum of life. Now, it depends whether they're in viruses or not, depends upon where you think viruses came from. Whether viruses were first cells that then stripped everything down and became, just optimized, reproducing machines. There are multiple views on where viruses come from. So I'm bracketing viruses entirely. But if you can recognize these kinds of phenomena, memory phenomena, non-associative learning, maybe even associative learning. If amoeba can do it, maybe microbes can too. Who knows? Maybe bacteria can do it too. We don't know. Decision making, error correction, those sorts of things. If that is happening at the molecular level, at the level of what we have always called physiology, which is what Brian Goodwin saw when he was working in developmental biology in the 1970s, and he was looking at enzymatic action. That's what he was seeing, and he put a label to it, and I think it probably ruined his career. He had a career anyway because he had a brain the size of a planet, but I think he was living that down for a very long time. I haven't been able to talk to anybody who can confirm that yet, but I'm not a biographer. I'm not a biographer, but I think that Waddington was dead. He didn't have his mentor and his defender there, and he took things a step further than even Waddington would have gone. And I think he just got chopped off at the knees. Myself, that's what I think happened. But so we can ask different questions. We can understand related phenomena much better than we can now, which will allow us to start developing the theories that biology really needs.
Mike, what are your thoughts?
Yeah, no, I think what you just said is critical because a lot of people think that this kind of talk is poetry, and that they say, oh well, sure you can say that, but what's the point? Okay, no, the whole point is, as Pamela just said, the judge of the criterion for all of this is that it has to lead to new discoveries, it has to lead to new research programs, and it has to lead to new capabilities. Otherwise, there's no point. All the verbiage, all the terminology, all of it is what does it enable? What does the old view constrain, and what does this view enable you to do that you couldn't do before? And that's very important. And just as an example, gene regulatory networks. Okay, just a set of a handful of chemicals turning each other on and off. So you look at these things and you say, okay, they're deterministic. They look pretty dumb and mechanical. And when that's your worldview, you're only and they're important for everything: health, disease, development, evolution, everything. When you make that assumption, it has a very specific implication. It means that if you want to change the way they behave, you're going to use dynamical systems theory, and you're going to have to rewire the thing. Gene therapy, you're adding nodes, removing nodes, changing promoters, physically rewiring the hardware is your only option. If you instead, as we did a few years ago, say that, well, you can't just assume anything is dumb and mechanical any more than you can just assume that, hey, there's a majestic spirit in every rock. Like maybe there is, but you have to show that that's helpful in some fashion, right? So if you do the experiment and you say, okay, we're not going to assume, we're going to do actual experiments. Well, it turns out these things can do 6 different kinds of learning, including Pavlovian conditioning. You don't need the cell, the nucleus, you don't need any of that. Just from the math alone, they can do it. And so that opens the opportunity for something completely different, which is drug conditioning, and that has huge implications for medicine and the way you use. And so we're now doing all this with real cells. But the idea is that you can then, if you don't assume where things are on that spectrum of persuadability, but you do the experiments, whole new sets of tools open up to you. And so all of this stuff that neuroscience has come up with, not only tools and techniques, but some actually amazing formalisms and some amazing tools for thinking about how collectives work and how multi-scale controls work and all this stuff. it gives you permission to now take those tools and apply them to things that aren't brains. Okay,
Yeah.
because I don't think neuroscience is about neurons in any case. And so right now you have the permission to do that. To do that. If you don't think that's the case, you will never be motivated to do that. You will never try these crazy things, and you will never try. You're not going to find the new biology. So I think it's extremely liberating, because by dissolving these old categories, you get to port tools and test them out and see what you can get. And this is why we put embryos on memory blockers and hallucinogens, and this is how you discover novel regenerative applications and everything, by asking what does the system know, what does it remember, what does it believe, and how do I convince it to do something different? Not force it at the molecular level and get all kinds of unexpected side effects as it tries to fight you on what you just did, but actually get buy-in from the system. And I think it's going to be massively important in many practical applications. And then hopefully eventually, my hope is that, I mean, Chris Fields has the saying, he says, only technology settle arguments. So I think we can say this stuff and then people will complain and whatever. But when the applications start coming forward, then eventually it all will tick over and everybody say, we knew it all along. Right. I mean, I'm kind of seeing this on the horizon already. Eventually he said, everybody always knew this. What are you talking about? Who needed you to say this? Everybody, we already knew this. So it just kind of like flips over to that. And then, it's fine. Yeah.
I just want to stress how groundbreaking some of this work is, because for most people, when they think of cognition, they think of neuroscience and they think of the brain. But both of you are totally right. When you start thinking of cognition in these different systems or cells, tissues, microbes, all the way down, it opens up such a different perspective. You can start, as you said, thinking of new theories. You can start thinking of new applications and just new experiments to run, which, Mike has done some beautiful work around that. And Pamela, you have some amazing work too. So I guess one question here is also, how do you start thinking about cognition in artificial intelligence then? If you guys have thought about that.
I can't do, that's not my area. I really can't do that. I have to stay with the living. I don't even go there. So Mike will have to speak there. But I will say that if the view that Mike and I are advocating was accepted, became accepted to the point where it was taught in schools to children, we would not be having the trouble we are having right now dealing with climate change. We would understand so much more about the commonality of everything that lives, and how much we rely on other living things and that sort of stuff. We wouldn't be having this trouble. So I will hand over AI to Mike.
Yeah. Well, let's see. I guess I can say this. As far as I can tell, so two things. So first, as far as I can tell, the architecture of life, that is what's critical about life, and we can talk about what that is, but partially it is this, it's the creative aspect of never being sure of what your memories mean, and having to constantly reinterpret your own engrams, because you don't have access to the past. What you have access to are the traces that past you, the messages that previous you has left for you, and you need to reinterpret them as best as you can. Right? Our computer technology is exactly the opposite. We have abstraction layers and error correction and redundancy codes and all this stuff to make sure that the data stay fixed. The data is because we are interpreting the data, and you don't want your registers floating off, and then you don't know what's going on, right? So life is not like that at all. So from that perspective, all of the things that we've made are not the right architecture at all. However, having said all that, one thing I've now realized is that if we don't even know what the damn bubble sort is doing, which is like 6 lines of code, totally deterministic, totally transparent, and we have now found that it does all these weird side quests while it's sorting the numbers. It also does all this other weird shit. If we don't even know what that does, we should be extremely cautious and have some humility around the fact that when you make that something, You do, just because you made it, that does not mean you know what it is and what it does. And I don't believe, when they say artificial intelligence, I don't believe we make intelligence synthetically any more than we make it biologically. I think what we make are pointers or interfaces that pull down certain patterns. And sometimes, if you're a good engineer, you'll pull down the thing you want. But very often, you pull down a bunch of other stuff that you did not expect, not just complexity, not just unpredictability, but actual cognition, emergent cognition. And so, we can look at these architectures and we can say, well, they don't even have a homeostatic feedback loop, or whatever. Yeah, well, maybe. And they're certainly not pulling down anything remotely like a human mind, or probably any kind of biological mind. But that doesn't mean that we're not currently fishing in some corners of that space that maybe have never been embodied before, probably not on Earth, but maybe nowhere else. And so I think we need a lot of caution about that. People have said, like, I've had debates with people. Somebody who writes these AIs will say, "Well, I make them. I know what they. It's just linear algebra. I know what they do." And I say, "Well, first of all, you don't even know what bubble sort does. And second of all, as it turns out, it's just linear algebra, and you're just chemistry or quantum foam or something. But of course you're not, right? So why is that? So why are we fairly comfortable to say that the the story of biochemistry is not the story of the human mind, but you somehow think that the story of algorithms and Turing machines is the story of these things that you're now making? Those are our abstractions with the limitations of the formal model. They're not necessarily limitations of the thing itself. So that's my story on AI. I think we have no idea what we're making, actually. Very, very, very little.
Well, you've just scared me to death, Mike.
That's my job. You know what? I was
a lot.
Yeah, sorry. I should have. I often apologize in advance. A couple of 2 weeks ago, Mark Solms had me at the Psychoanalytic Association. I was on Zoom giving a talk to the psychoanalysts, and that's the first thing I said, because they wanted to hear about it. And all this. I said, I'm sorry, because what I'm about to tell you, I'm sure is not going to make you feel better. You're gonna have to really put your thinking cap on and ask yourself how you're going to offer your services, which are very needed, to a very wide range of clients in the future. Things you can't even imagine: hybrids and cyborgs and novel beings of a variety that Darwin's endless forms most beautiful didn't even begin to scratch that the surface of. So
Pamela, I know you approach this work coming from Buddhist philosophy, and you still do a lot of work with Buddhist monks and Eastern philosophy. And it actually strikes me that some of the views on cognition are more accepted by Eastern philosophy. It may not be from a very scientific perspective, but I think people sort of embody that in many ways, if you know what I'm getting at. Do you think that's true? Could you maybe explain that a little bit? And why do you think, I guess, the East maybe accepts it more compared to the West?
That's a great question. Thank you. I will, how to say, okay, you have to understand first and foremost that I am a heretic. Okay, I am a heretic. When I undertook this PhD in this project, I had to risk losing my faith, but I didn't. But unfortunately, I turned up things that my particular tradition doesn't accept. Do you know what I mean? They don't accept plants. Bacteria, are you kidding? Molecular, the workings of the body as being cognitive, are you kidding? So there's an entire layer of what I do that is not acceptable to the Buddhist view as it stands right now. Now, it was more labile in the very, very early years of Buddhism, but in certain Mahayana traditions, it really, really hardened. It really hardened. And, but the idea that all living, because they use the term living, all living beings, which meant that plants had to be put in another category, that they weren't actually alive. So in my lifetime, I have had educated, not Western educated, but educated, highly learned geshis in the Tibetan tradition sit up on the teaching throne and describe how plants aren't alive. I've had to listen to that. I've had to listen to that and try to correct them, but can't be corrected, right? Or it's like kind of like la 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 la. Let's just not talk about it. Let's not talk about it. And but once the Buddhists decided that living things were minded creatures, they were cognizers. There was something about being alive that they saw as being associated with cognition, which is why they could see it in insects and really, really, really small things like that. And there was no problem in seeing cognition in an ant and a human, right? And that came out of the incredibly rich tradition of classical India, of the Rig Veda and classic Hinduism, and then the reactions to classical Hinduism by Mahavira, who was the founder of the Jains, and Buddha, who was founded Buddhism, and they reacted against those pictures with their empirical experiences. Do you know what I mean? They did an investigation and they reported on what they found, basically, rather than relying on scripture. But all of that came out of very, very, very rich tradition in India, and that spread everywhere, as William Dalrymple mentions in his latest book, The Golden Road, is that Buddhism was probably one of the greatest exports that India ever had. I mean, it went everywhere. It went everywhere. It went to the Roman Empire. It went to Greece. It went to China. It went to Southeast Asia. It went everywhere. And it profoundly changed the way people think. So people in the East basically have that view because of classical Hinduism and then the traditions that arose within that tradition. And one of my favorite stories out of this is in 1983. This was 1983. Don't forget that. Don't forget this was in 1983. Imanishi, the eminent Japanese primatologist, had been publishing work on Japanese macaques saying, we think we're seeing culture here. We think we're seeing cultural transmission in Japanese macaques and certain ways of doing things that are different between different troops, right? They're different between a scientist, a paleontologist from England, from the UK, was so outraged by this. He flew all the way to Japan and tried to talk Imanishi out of this, that this is insane talk, can't possibly be true. Couldn't convince him. Came back, was given 3 pages in Nature to trash Imanishi's entire approach to primatology as anti-Darwinian, childish, all this sort of stuff. And we don't know about any culture in animals now, do we? We have culture everywhere in animals. We have it in birds. We have it in cetaceans. We have it in all kinds of animals. And Nature gave this guy 3 pages to trash the entire idea. What was the difference between Imanishi and Halstead? Imanishi came from a Buddhist culture, and Halstead came from Judeo-Christianity. Good question.
Yeah, I'm actually Jain and grew up with Jainism, so it was part of the reason I asked the question. It's also just intuitively easy for me to grasp. I know, Mike, you have to run, so.
I will run in a minute.
But see, Mahavira believed in plant cognition.
Mm-hmm.
Mahavira, the Jains believe in plant cognition, but the Buddhists did not.
That's right. Yeah.
Yeah. Just a very quick anecdote along those lines. I've talked to people at the Vatican. I've talked to rabbis and whatnot, and that's you kind of get what you expect. But I gave a talk once at a I'm not going to call them out by name, but it was an an Indic kind of tradition. And I gave them a talk, and I thought, okay, these guys are going to love this because finally there's a scientist that's saying some of this stuff. Well, they flipped out because they were so mad. They were so mad that I had the non-living things on my spectrum, and they kept saying, no, absolutely not. They said it's dead matter. I said, what? There is no dead matter. There's just like lazy observers and things like this. What makes you think that, And they said, no, absolutely not. And I said, aren't you the guys that talk about the soul, incarnating in bodies? And they said, well, yes, living bodies. And I said, well, who are you to tell these spirits if you make a beautiful, robotic embodiment that somebody's not going to come live in it? And by the way, I've heard people talk about, you might get reborn as a block of wood and all this kind of stuff. And you're telling me now that they're not going to come live in these, in these synthetic embodiments. I say absolutely not. I mean, they were just so incredibly attached to this sharp separation. They wanted the dead matter, and then they wanted like all this awesome magical stuff. And it just, there was just no getting past it.
Fascinating. All right. Mike, any last questions, thoughts?
No, thank you so much, Pamela. It is always delightful to see you. You're an inspiration. I've said it before. The stuff you've done way ahead of the curve of anybody in this area. So just amazing.
Hi everyone. If you wish to be notified of future podcast episodes and my writing on longevity and the frontiers of biology, please subscribe at livelongerworld.com. And if you're enjoying the show, please leave a rating on Apple, Spotify, or elsewhere. Thank you for listening, and I will see you next time.